Hello there. Welcome back to the 5th episode of my Foundations of Neuroscience series. Sorry for missing the last upload. I got sick and got caught up behind a bunch of other commitments, but hopefully I should be back to schedule after this one. The last couple times, we looked at action potentials and graded potentials, and today we're going to see what happens when things end up at the end of the axon. Once the axon potential reaches the terminal, the signal needs to be transferred from one neuron to the next. This is done at the synapse. By the end of this video, you should be able to answer the questions: what are the different types of synaptic connections? And how do axons communicate through synapses? The two main types of synapses are electrical and chemical. Electrical synapses are really easy as the current is just carried across from neuron to neuron. The signal passes through structures called gap junctions. These allow really quick connections and are ideal for when you need tight synchronicity with neuronal responses. However, the more common synapse for us is the chemical synapse, where a class of molecules called neurotransmitters are used to convert the electrical signal of an action potential into a chemical one. Once the neurotransmitter makes it across to the postsynaptic cell, another graded or action potential can be triggered. Remember back when I introduced the stages of an action potential and I mentioned how the cell would have to move from rest to threshold? This is what tells the cell to do that. The downstream results of a neurotransmitter binding enables the cocktail of different responses. Alright, quick comprehension check with the question. Electrical synapses communicate through which structures? A. Gap junctions. B. Nodes of Ranvier. C. Axon. Or D. Soma. No tricks here, it's just answer choice A, gap junctions. The other answer choices are other random things in the neuron. The classic example of a synapse sees the connection between an axon of the presynaptic neuron and the postsynaptic dendrite, but that isn't the only way a synapse can manifest. You can get axons joining straight onto a soma, axosomatic, another axon, axoaxonic, or a non-neuron, like we see in muscles. A synapse can even land on the same cell, something called an autotaps. In most humans and other vertebrates, most synapses are unidirectional, where it goes from pre to post, but there's always exceptions. So, if electrical synapses are faster than chemical ones, why do we use neurotransmitters for synaptic transmission? The answer lies in the immense variety in neurotransmitters, which can be broadly grouped into amino acids, amines, and neuropeptides, which are small proteins. There are some other classifications too, but there's no need to go into it right now. Despite the different classifications, neurotransmitters share some key properties. Namely, neurotransmitters are going to be molecules that are synthesized, packaged, and then released into the synaptic cleft, and then are subsequently broken down either by uptake or just enzymes in the cleft. Now that we know where synapses are and what they release, it's time to talk about how neurotransmitters are released. The neuron uses synaptic vesicles that are chock full of neurotransmitters, dumping them into the synaptic cleft, and then letting them diffuse over to the other side where they can bind to their relevant receptors. The simple explanation of neurotransmitter release by vesicle fusion is this. The vesicle can get close to the membrane with something called a snare complex. Once the action potential reaches the terminal, voltage-gated calcium channels will open up, allowing the calcium to rush into the cell. Calcium influx acts as the trigger for vesicle fusion, pushing the neurotransmitters out into the cleft. Ligand-gated receptors specialized for the neurotransmitter can bind on the postsynaptic sides, causing some response. A neurotransmitter's effect is based on the receptor on the other side of the synapse. If there isn't a receptor on the other side, well, nothing will happen. Neurotransmitters can be inhibitory or excitatory. Excitatory neurotransmitters help trigger action potentials in the postsynaptic cells. Common examples are glutamate and acetylcholine in your muscles. An example of the mechanism of this would be glutamate binding ion channels, whose opening allows cation influx, thereby depolarizing the membrane and bringing it up to threshold. On the other hand, inhibitory effects are commonly seen with things like GABA and glycine. Sometimes the same neurotransmitter has different effects on different types of neurons. Acetylcholine is the reason your muscles move, functioning in an excitatory context, but in some parts of the brain, the very same acetylcholine molecule has an inhibitory effect. While these neurotransmitter-gated ion channels, aka ionotropic receptors, are useful, they're more short-term. Longer-term effects are usually carried out by metabotropic receptors, which, upon binding, can cause chain effects in pathways that can even upregulate the number of receptors in the postsynaptic membrane themselves. Alright, time for another question. You discover a new receptor that binds GABA. What is the most likely effect and mechanism of this receptor? A. Excitatory with cation influx. B. Excitatory with anion influx. C. Inhibitory with cation influx. Or D. Inhibitory with anion influx. Give you a second. The answer here is D. GABA is a stereotypical inhibitory neurotransmitter. This eliminates answer choices A and B. If we wanted to inhibit firing, we're going to want to make it harder to get to threshold. This is hyperpolarization. Cation influx makes the membrane more positive. That's depolarization, so answer choice C is wrong. This leaves the answer choice D, where the negative charge of anions entering the cell hyperpolarizes it, causing inhibition. Well, that's all I have for today. We covered a lot about the mechanics of synaptic transmission, looking at where, how, and why of synaptic transmission. Next time, we'll look at how these signals are used for information encoding. As usual, resources and timestamps are in the description. Once again, sorry for the delay on this one, and thank you very much for watching. I hope to see you next time.